All right, let's do chapter 7. Chapter 7 has the signaling. And so we're going to take our time and we're going to go through the different signaling cascades. We're going to be helped out. There's the dog back there. All right, he's going to help us out. Let's go. Let's do this. This is learnable. Okay, as always, these are for you, and I'm going to skip them while doing this, but these are useful little definitions and helpful little things, I think. All right, so here we have CD3 and CD3 and the T cell receptor. Yeah, there's no CD4 in there, but it's coming, in the next one. So just looking at this thing, this is what we described in class. So we've got our actual T cell receptor, which is the blue thing. This is the thing that's built like an antibody. And a T cell receptor, it just sticks to something. It sticks to MHC with a particular peptide on it. And yeah, if it makes the right T cell receptor, it can stick to that particular peptide. If not, it just won't stick. So there's a lot of trying and failing to bind, and that's the T cell's life. All the other chains you see around here, so this gamma, delta, epsilon, another epsilon, and the zeta chain down here. That's what that little curly Q looking thing is. These are all together CD3, basically. Some people would say maybe just these four are CD3, but they all basically constitute the things which are always found around a T cell receptor. So call it all CD3. All of these are signaling components. So each of these yellow boxes is going to be a spot that has some tyrosines in it. And those tyrosines can be phosphorylated to make a kind of a docking site. It's kind of a hub where other protein kinases can land and then can interact. If you're interested in the pluses and minuses, I think these are charges in the hydrophobic transmembrane region. There's like a little charged amino acid. And so the charges are what sort of sticks this together. I believe that's what they're getting at there. So that's CD3. And so CD3 doesn't have an alpha and a beta chain. It just has gamma, delta, epsilon if you're, one of those alphabet types. The alpha and the beta come from the alpha beta T cell receptor. There you go. All right. One of the major proteins that is going to bind to all of those signaling domains is going to be called ZAP70. And so here's ZAP70. So wherever you see something that looks like a tic tac, just like this, a little oval with a thing carved out of it, like somebody took a little bite out of it, that is going to be a protein kinase. And so you want to watch for those. That's another one of those symbols that every time it comes up in the book, it means the same thing. And so even if you can see it, it'll stick, you'll remember it. This over here is called an SH2 domain. Now those are neat. SH2 domains are able to bind to phosphotyrosines. And this actually has two SH2 domains right next to each other. So it can bind two phosphotyrosines, just like that. And the other neat thing about ZAP70 is that when it is closed up like this, sort of the starting conformation, it's not a kinase. It can't work as a kinase. It is inactive. The thing that activates it is getting that SH2 domain away. So the SH2 domain is sort of crunching this thing together. And it's just like a pocket knife. When it's closed, you can throw it at something, but it's not going to do much. Yeah. When you open it up, that's when it can start to do things. So we start out with ZAP70 here, inactive, and it's going to get activated by binding to tyrosines that are phosphorylated. Now, ZAP70 didn't necessarily phosphorylate these tyrosines. Something else did. So we'll solve that mystery in, about 3 slides or so. Wait up for that one. Don't go to bed yet. Every time you see a little red circle in here, that is a phosphate group. So that is just how this book does it, and it's super consistent all the way through. You can see I pulled up a diagram. You see, this is the same thing. This is that T cell receptor complex. And instead of drawing ZAP70 in a way that makes sense, they've just drawn as a big circle, which is easier to draw, but less useful overall. It doesn't tell you how a thing works. And I'm super interested in how this stuff works. Because if you understand what's going on at the level of how different parts of different proteins work, then you can understand when something goes wrong. Okay, you can say, all right, this part's broken, therefore it connects to this part, and in a knee bone connects to the leg bone kind of way, you can figure out exactly what's going to happen. It's like if you pull out a particular Jenga brick, you can tell, which one's going to topple the tower. It's like that. All right. So, next, let's go to this one. So remember B cells. They have that thing called the B cell receptor, which is basically a surface-mounted IgM or IgD molecule. IgM being the first one, and then IgD being spliced, from that, differentially spliced. So that part we know. And antibodies don't actually signal by themselves. So we again have to add some signaling molecules to detect when the antibody has caught something and then to signal. Yeah, so we basically functionalize the T cell receptor and the surface-mounted antibody, the B cell receptor, by adding these domains. These domains have the yellow squares with the tyrosines. Those are called ITAMs. And that's on a previous slide, I believe. Let's go back up. Here we go. Immunoreceptor, tyrosine-based activation motif. So an ITAM is something that is always going to activate the protein when it gets phosphorylated. And it's always going to have a tyrosine. That's the thing that'll get phosphorylated. And it can have two. It can have more than two. Actually, ITAMs tend to have two. It cannot have more than two. Sorry, got carried away there for a sec. So they'll have two tyrosines that can be phosphorylated to make this double phosphate pattern that SH2 domains can bind. And you find SH2 domains on lots of proteins, and those are the proteins that are going to be able to bind to an activated T cell receptor complex. They'll be binding to some of those CD3 chains that are phosphorylated. That's just how it works. So immunoreceptor, tyrosine-based activation motif. So every ITAM is activating. Now, there's a different kind of tyrosine-based motif that inhibits instead of activating. So if it is a tyrosine-based motif that will inhibit some enzyme, then that's going to be called an ITIM, immunoreceptor tyrosine-based inhibitory motif. Yeah, we'll get to those later, but mostly you're going to see ITAMs, and ITAMs are those yellow rectangles, so watch for them. Yeah. All right, look at all those ITAMs. Okay, and so when it says ITAM down here, you don't even need it to say ITAM, because you can see, oh, it's a rectangle, it's yellow, it's an ITAM, and it's in the tail of a protein next to one of these immunoreceptors. Yeah. So that's going to be something that is going to get phosphorylated. And once it's phosphorylated, it can serve as a docking place for ZAP70, because ZAP70 needs two little phosphates on an ITAM. That's its deal. That's its binding selectivity. And then once it has that, the SH2 domain will basically let go of the kinase domain, and the kinase domain can start to work. Yeah. And that's it. That's how all these things work at a molecular level. Something's always in the way, and then when you phosphorylate or whatever, bind to another protein, then it moves out of the way, and then all of a sudden the protein can do the job that it's supposed to do. Yeah. All right. So let's go. Fine. So here's how it works. And this is the overview, and believe me, we will go through every part of this. Yeah, but overview time. So this is a helper T cell, and it can be any one of those, Th1, Th2, Th3. The only ones that sometimes do not have CD3, I believe, are T regulatory cells. They would be low in CD3 production normally, if I remember correctly. That's, not going to be on a test. Let's see. So CD4 coming in here, so you know it's not a cytotoxic T cell. And so, of course, we're going to bind MHC class 2, which means that this is an antigen-presenting cell, probably a dendritic cell or macrophage, but when we get a good old inflammatory response going, it could be a lot of different cell types, just a little bit. They're not They're not great at it, even under extreme duress, but they can do a little. Bless. Yeah. They do their best. Okay. CD4 is associated with a kinase of its own called LCK. And so I imagine this is like a deer at a salt lick. Lick kinase, or you can call it luck kinase. You can think, oh, that's lucky. When CD4 binds to MHC class 2, the only way that happens is if MHC class 2 is held in place with a T cell receptor. Otherwise, you'll get CD4 binding or you'll get T cell receptor binding, but no CD4, and this only works if you get all those things bound at the same time in the same place. So, when CD4 binds, it's going to bring in LCK. And LCK is a kinase. And so the first thing LCK is going to do is it is going to add phosphates to all of these little ITAMs. Boop, 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 boop. Just like that. And then once there are phosphates on the ITAMs, you know who's going to bind? ZAP70 is going to bind. And once it binds, it's going to then go on and add phosphates to other things. That is the idea, and that's how we're going to start the signaling cascade. And we'll go a little way into the signaling cascade, and in some cases we'll get all the way to the nucleus, but yeah, that's where all signaling cascades end, basically, is some signal being sent to the nucleus. And by signal, I mean a transcription factor, like a little protein that's going to bind DNA like a bookmark and tell the cell, right here, make me one of these, please. Don't know what it is, but I feel like we need it. So there are lots of different transcription factors, and they are going to be deployed in teams. Like when a cell receives a little bit of interferon, it's going to send out its interferon response team. And so there are going to be all these interferon stimulated genes. And it'll be the same transcription factors running most of these genes. But then, yeah, sure enough, you'll get expression of a whole program. You know, you can have 100 different genes come on when you receive one signal. And that's really neat. There's a way inside the cell that the signal sort of amplifies. And so a cell can make a big response to a small stimulus, which can at times be a lifesaver. It sort of speeds up the process, perhaps. Okay, so let's look at these. All right, fine. So just starting out right here, how does this work? Now, CD4 and CD8 both have two cysteines in them. And cysteine is one of those amino acids that has a sulfur near the end of it. And the thing about these sulfur amino acids, that you know about disulfide bonding, right? Where they have a sulfur and then a hydrogen, they kick off their hydrogens and they bond to each other, just like that. Nice covalent bond. That's one thing that they can do. The other thing you can do with these disulfide making type guys is if you have four either cysteines or histidines, you can actually surround a zinc ion. So it's a tetrahedron in 3 dimensions, if you can imagine that. And so, yeah, that's what they do. So it takes 4 of these, and they will coordinate one zinc atom. And the zinc atom basically holds this protein over here to this protein. Really, it's holding these cysteines to these cysteines, and these are attached to a CD4 or a CD8, and these are attached to the LUK kinase. LUK. The K is for kinase. That's redundant. Yeah. So, to be clear, both CD4 and CD8 protein have this. So it'll work the same way on a killer T cell, except we'll have CD8 and we'll be interacting with MHC class 1, but otherwise it's the same deal. When the co-receptor comes in, CD4 or CD8, it's going to bring with it a kinase, and the kinase is then going to activate the T cell receptor. Yeah, so that's it. That's how the binding works. I mean, none of it's particularly complicated, and probably wouldn't work if it was complicated. It's just, yeah, it's just the way this happens. So, now look at this. We got 2 domains here that you've seen, and 1 domain that's new. So, domains you've seen. We got a kinase domain, and you can totally recognize it. Doesn't matter. They'll sometimes put the little notch up near the top. Sometimes they'll put it in the middle. Whatever. And they'll change the color on it to show that it's a different kinase, but you recognize the shape. That's what tells you it's a kinase. Yeah. So at the start, you've got the LUC kinase, and basically it's got one SH2 domain. So it doesn't have two like ZAP70, it just has one SH2 domain. And it binds to one phosphotyrosine on the tail of LUC. And what that does is basically it's like trying to scratch your own back or something like that. It's just locked basically in that position. So the kinase doesn't work because the tail is being held in place by the SH2 because of its interaction with that phosphate. So what needs to happen is we got to find some way to get rid of this phosphate and turn it back into just a tyrosine with no phosphate on it, which they abbreviate as a little Y right there. And when that happens, then that is going to be the signal that, basically starts the process off. So the enzyme that does this, so an enzyme that adds a phosphate is called a kinase, confusingly. An enzyme that takes away a phosphate is a phosphatase. So phosphatase breaks phosphate, takes phosphate off of a protein. And there is a phosphatase that is also going to be part of this complex, and it's called CD45. So there are different types of CD45, but I believe they both come in with the right equipment on the bottom. So CD45 has a phosphatase attached to it, and CD45 will take off the phosphate from LUC if it's there. And once that is taken off, now the kinase domain can work, and the kinase domain is going to go over and first, it phosphorylates itself. That's what you call self-care. Second, it phosphorylates the ITAMs. It just goes along, and every ITAM it can reach, it just pops a phosphate on there. And that's just the standard reaction. You take, I believe it's an ATP, and smash it, and you drop off an ADP and stick the phosphate on. It might be a different nucleotide triphosphate. I don't remember which one this particular kinase uses, but yeah, that's the idea. That's the idea. Okay. Oh yeah, and ZAP70's name is zeta chain associated protein, and I think it's 70 kilodaltons, something like that. So that's why it's got that name. All right, so this is what a ZAP70 looks like, and drink it in. So you remember ZAP70? ZAP70 is going to have a SH2 motif, and so that's our green one here and our blue one. So these are both SH2 motifs, and it looks like they're not attached, but that's just because of the way the crystal structure is solved. Bits that are a little bit floppy are not shown. Just because you can't tell exactly where they are. You can tell roughly, you know, it connects here and here, but it could flop around a little bit in between, like that. So this is all part of the same protein, and you got your SH2s. And while these SH2s are interacting here, basically it is closed, and it is not active. This big orange domain is the kinase domain, and the two little red loops here are two tyrosines on there. So LUC, LCK, is going to phosphorylate these two tyrosines right here, and then that's going to start the process. So that's where we're going to pause this one, and then we're going to go more into LUC and CD45 and ZAP70 and what happens after in the next section. So I'll be right back.